hope you can hear me well. Thank you for joining today in this special webinar. Today is our first formal event as part of the Integrative Conservation webinar series. These webinars will happen once a month, the last Tuesday of every month at this time, 4:30 to 6 p.m. Beijing time. And they are part of the weekly program of the XTBG webinars. So then XTBG has one webinar every week, and the last of every month will be sponsored by Integrative Conservation. I hope by now you have heard about Integrative Conservation. It's a new academic journal launched by XTBG. It was launched in December, just 1 month ago. And we published the first issue, and I hope you have seen it and you had chance to browse the content, and you are considering submitting your papers if you work in conservation. So then today we have a very special guest speaker, Professor Harald Schneider. Professor Schneider is the PI at the Macroevolution Group in the Center for Integrative Conservation at the XTBG at the Xishuangbanna Tropical Botanical Garden. Professor Schneider's research focuses on the genomic mechanism shaping the evolutionary history of land-plant diversity since the colonization of land and their survival in the Anthropocene. He promotes integrative research approaches with the aim of incorporating state-of-the-art research together with the knowledge accumulated during centuries of botanical exploration of the world. Professor Harald Schneider has a very fruitful career, and he has contributed to many important publications, reported results that are now part of every textbook on plant taxonomy and diversity. He got his PhD at the University of Zurich and has served as a faculty in many prestigious and famous institutions, including Leiden University, George August University at Göttingen, Duke University at Durham, University of California, Berkeley, and the Natural History Museum in London before joining XTBG. Professor Harald Schneider, according to research.com, he ranks among the top 20 experts in ecology and evolution in China. And he will be talking today about his work. I want to highlight that the presentation he's giving today corresponds to a paper that he is publishing in the March issue of Integrative Conservation, and the paper will be online in 1 or 2 days. So then I encourage you to check the website in a few days and see the early view version of the paper. The title of the presentation is Integrative Genomics and Conservation to Safeguard Plant Diversity. And Professor Harald Schneider serves as an Associate Editor in Integrative Conservation. So thank you very much, Harald. And now the floor is yours. You can proceed to share your slides and go with the presentation. Just for everyone, Professor Schneider will do presentation for 45-50 minutes, and then we'll have discussion. So then please stay for the Q&A session after that. Thank you, Harald. All yours.
Thank you, Ahimsa. And I will share my screen now. And now it should be going. Okay. Welcome, everyone. Good afternoon, good morning, or good evening, wherever time zone you are in. It's a great pleasure to give the first of seminars of the new seminar series, Integrative Conservation. And it's a very large honor. And thank you very much, Ahimsa, for organizing and everyone else involved in organization of this seminar series. As Ahimsa mentioned already, I will talk about the content of a manuscript which is now in press, which will come out hopefully as soon as possible. And I was very pleased to hear it's going to be published in 2 days. Which will be published in Integrative Conservation, which focus on integrating genomics and conservation to safeguard plant diversity. In my presentation today, I will have special reflection on genomics in the context of the global biodiversity framework. And I think it's very important that all speakers in the seminar series reflect very carefully what the global biodiversity framework means for conservation in their context of their sciences. Here in China, we only came back from the celebration of the Chinese New Year, so I will take the advantage to wish you all a happy new Chinese New Year, the year of the rabbit. And I also want to celebrate one other thing. Therefore, very soon we have the International Darwin Day, where we celebrate the birthday of Charles Darwin, which was born on the 12th February 1809. And my talk is part of this International Darwin Day, despite a little bit too early, only 12 days too early. But it is very important to celebrate Charles Darwin. If we think about the global biodiversity framework, actually humanity is in a major challenge ahead. We need to reduce our footprint. Our footprint is now so heavy that we talk about a new century, geological century, which we call the Anthropocene, the century of humanity. And one of the big issues of the century is biodiversity crisis, a large amount of species which are threatened to be going extinct. But the 21st century is also the century of what we often call the post-genomic area, meaning we have now full access to the genome blueprint underlying all the organisms. And this new perspective provides not substantial innovations, and these innovations are very critical to achieve the goals set up by a global biodiversity framework. When you look to the text of the global biodiversity framework as someone interested in genomics and genetic diversity, you are very pleased to read the text. And for example, the 4 goals for 2050, goal A mentions specifically the maintaining of genetic diversity in wild and domesticated species, and set up a very ambitious goal with saying at least 90% of the genetic diversity within all species maintained. This is really a very ambitious number. And the focus on genetic resources continues also in goal C, which talks about benefit sharing of utilization of biodiversity by mentioning clearly genetic resources. If you go further in the text and look to the milestone to be reached by 2030, you have to wait to milestone A3, where actually genetic diversity of wild and domestic species are again clearly mentioned. And again, we see this target of 90% of the genetic diversity. Are we really ready to take that challenge that we have to preserve 90%, or we aim to preserve 90% of the genetic diversity of all organisms on Earth? When we think about it, we are talking really about big data, a huge amount of information which needs to be created, managed, and maintained. For biodiversity research institutes, this means also that we move biodiversity research from small lab or individual lab group research towards large infrastructure research. This is actually really a challenge we have to take on, because now we record biodiversity not anymore as from species number, but we have to record this in number of genetic diversity per species. This means that we have to address urgently some major gaps in standardization of protocols to record genetic diversity, and the infrastructure to carry out recording and maintaining this data is actually really urgently the need to build it up. Think a little bit, maybe we need to look back a little bit what actually this means in reality. And I want to illustrate this a little bit with numbers using the focus on Xishuangbanna diautonomous prefecture. That's where XTBG is located. It's a very known area for high biodiversity and has been explored very carefully. And therefore, I can now list, based on the exploration for all land plant villages, here starting with hornworts, liverbirds, mosses, lycophytes, ferns, gymnosperms, and then breaking the endosperms in major lineages, I can record very clearly the number of species which we have in this area, which is accumulated a total number of about 5,000 non-plant species occurring in Xishuangbanna alone. We can assess easily now, quantify the contribution of Xishuangbanna to the global biodiversity, which is in total 1.6% of the non-plant diversity occurring in this area. And you see in some groups we have more contributions than in others. I could also do this now for threat assessments, which would be actually also possible to do. But now with a new global biodiversity framework, we have to think about, can we actually quantify that also given the genetic diversity which is actually maintained in this area? And actually, to say at the moment, we wouldn't have the data to do that. For a couple of species, yes, but not for all of them. Saying that one is a challenge, for conservation biology will be in the post-genomic area increasingly driven by to protect genetic diversity instead of thinking only about single species diversity. Therefore, we have to do arguably a little bit catching up with other parts of biology where genomes actually drive into business already. If you go in the area of crop sciences, many of the important crop plants have been published genomes as rice, tomato, potato, corn, all the name some. And now these genomes already start to contribute to secure the food supply in the future, also under the condition of global climate change. And actually, we as biodiversity scientists working on conservation, we may have to think in the same way. James Watson, when one of the discoverers of the DNA, would once a book which he says DNA is the secret of life. And I personally would say when we have now full access to genomes, we can read the genomes, we can read the secret history of life. We can look at really how history evolved and under which mechanisms they evolved. I mentioned here a couple. And use as adaptive mutations through genome duplication, conservation of genetic pathways, the innovation of new genetic pathways, loss of function, loss of genes, refunctionalization of genes. All that questions can be answered and studied now with genomes. Some of you may actually think when I talk about conservation biology, post-genomic, think about Jurassic Park. Can we actually revive extinct species? Personally, I would say this is a little bit still a dream, but maybe possible dream. Personally, I would also argue we should actually focus more on the revival of species which are nearly extinct. Applying genomics to plants which are surviving in botanical gardens. So here, I want to give one example for such a plant. The St. Helena redwood, Trocheotopteris erythroxylon, which is extinct in the wild. All the surviving specimens are descendants from a single tree found on St. Helena Island in 1950. You have to be aware that this tree was surviving long after the major extinction happened for the species on St. Helena, which was actually driven by the seafire during the time of major discoveries where St. Helena was discovered. This is more than 100 years ago. And this individual has substantial problems with the reproductive biology. Therefore, it's not easy to revive that species without genetics. In this case, rescue genetics may be needed. Genomics are now widely actually already in the ex situ or in situ conservation of threatened species. Here I put a couple of species which have both the genome available and are managed carefully under conservation management protocols. Now, example our here from Xishuangbanna, which we published last year together in a larger collaboration with colleagues in Guangzhou. We looked to Adiantum nelumboides, which is a nice ornamental plant, which is endemic to the Three Gorges region in southern China, which is highly threatened by habitat transformation and therefore listed in China in the National Key Protected Wild Plants list. We recovered that species in polyploid genome, which actually has a big advantage for the conservation in this case. And having the whole genome available will help us in our efforts to ex situ conservation of that rare species and bring it back and maintain it. Therefore, basically, when we think about conservation part in the post-genomic area, to take two strategies to implement. The first strategy I actually talked about with some examples already. Look into highly threatened species, sequence their genomes, which can be then be utilized in ex situ and in situ conservation management plans. But we have to think maybe also on the second topic, using focus on the genomes of key organisms of selected environments, such as the tropical forest in southern Yunnan, which will be then support our efforts to restore ecosystems. And that one will require much more attention to genetic diversity restoration. This talk here is not somewhere in the empty space because some colleagues of us have very visionary projects already established. And I mentioned here 3 projects which think about the genomes of the tree of life. The central one, the Earth Biogenome Project, actually has specifically mentioned the goal to sequence all organisms alive, to protect this information from the future. The Darwin Tree of Life, which is driven by the Sanger Institute at Cambridge, has actually set the goal to sequence 70,000 species occurring in the British Isles, UK and Ireland, to maintain and safeguard this diversity. Another interesting project in this context is the California Conservation Genomics Project, which actually has the aim to build up the most comprehensive genome data set ever assembled for conservation science, by focusing specifically on endangered plants in organisms in California. And you know, California is very known for this high amount of endemics because of the unique geological history of California, which is now under threat by global climate change. All that doesn't come out of the blue, and it reflects the major advancements in plant genomics or genomics in general. We need to keep in mind that the first complete sequence plant genome was only published in 2000. This was the genome of Arabidopsis thaliana. In the time since, so roughly 22 years, we published more than 1,200 plant species have at least one genome published. Some have actually hundreds of now genomes published. This is a remarkable process, and which provides us huge advantages. In 2002, I published together with some colleagues of mine a paper which we talked about how to decide which plants should be sequenced, because we were so worried at this time, based on the costs and efforts, that most of the genomes published will be economically important plant. Yes, still we have a bias that more economically important plants have been sequenced. But if you look now to this wish list we had proposed in 2002, nearly all, the vast majority of these wishes have been finished already in 2022. And a couple of missing ones will be very soon be finished. Therefore, it looks rather remarkable, successful. We can actually look to the success also by number of species with a whole genome made available since 2000 per year. Starting in 2000 with Arbitraris Italiana, and you see actually each dot then how many species have been published per year, and you see that actually follows roughly an exponential growth curve. And this is really in this we have no reason to believe that this speed will decline. Actually, potentially gets faster because the cost goes down and the technical improvements make it easier. And therefore, we'll get a rapid improvement of data available. As a phylogenetist, I'm of course very concerned that which branches of the tree of life have been covered. Therefore, I try to illustrate this one here by a consensus of the land phylogeny with using odors as terms. Therefore, if you go through the thing, you see the hornworts and liverworts, mosses, lycophytes, ferns, gymnosperms, and angiosperms. And if the order is printed in green and has a number and part of this behind, this means that we have at least one species per order in the whole genome sequence. In some cases, as Solanase, we have now 60 species, Lamiated 65. So for in some cases, it's a very high number. But we have also orders which have no data yet, with some in angiosperms. But in general, the orders we have no genome sequence are nested in the non-seed land plants as mostly the liverworts and the mosses and basal ferns. Therefore, we still have work to do, but actually amazingly good, given that this has been achieved in 20 years. Given here the interest how we do actually in the context of threatened species. And therefore, to evaluate, I've done one simple calculation. I went through the database of the IUCN Red List criteria database and looked how many of the species, how many species would be proportionally extinct, threatened, or least concerned when you would evaluate all the taxa which are accepted in the database of the IUCN Red List, and then done the same stat analysis for the proportion if I look to the data taxa which have a genome available and assessed in the IUCN Red List. And what you see is that, not surprisingly, non-threatened species are overrepresented. This is this purple color. But at the same time, the positive news is also a lot of critical endangered plants have assessment whole genome made available. Therefore, it's actually rather positive what happened on it. We talk here about reference genomes, meaning genomes which are fully annotated, but there's still the number is limited. 1,200 versus 3 million plant plants is not a good balance. And as I showed, we have some bias in the phylogeny distribution and bias towards unthreatened species. However, genomic application of genomic approaches is not required to have a reference genome for the species already available, but we have to keep in mind that analyze get more powerful if the same species or closely related species can be utilized, have a genome that can be utilized. Okay, let me come back to the two strategies I'm talking about. And I want to now focus a little bit more on the scientific need to think about why we do that. For that one, I have to detour a little bit to the genomics or population genomic approaches. If you have population size, and the population size declines rapidly, which is typical for threatened species, your frequency of inbreeding increase, the genetic drift effect comes in, and in total, the genetic diversity declines. At the same time, most threatened species experience change in the environment, means a highly stronger selection, which actually interacts with the same time with a reduced fitness because based on the reduced genetic diversity. And this actually enhances the extinction threats. I made a little bit more complicated model now here. I assembled this recently, and this is now in a review, which I call evolutionary genomic extinction model, which integrates population genetics and ecological understanding of extinction processes. If you have a species which have a large population, that is in the center of this model, this ST area, and which should be stable, then you have a lot of individuals, and the competition among individuals of the same species is high, but also the outbreeding frequency is normally high. This means also the robustness of the species against environmental change is normally very high because you have more genetic diversity to adapt. Now we go to species which have a population decline as a consequence of change of environment. This means that the population size reduces, meaning in the same time that the interspecies competition is reduced and the inbreeding frequency gets increased. In turn, that's the most important part to realize, the genetic diversity will decline in the species. And decrease of genetic diversity means at the same time you have an enhanced vulnerability to environmental change because you have less genes to adapt to the new conditions. Therefore, that's actually really bad what actually happens. And that when you try to return species that are close to extinction, this is one of the challenges we have to take on. But actually it gets more fun. We have to think about what we could call the genomic meltdown or genomic erosion in threatened species. As I said, we have a rapid decline of population size, increase of inbreeding, reduction of intraspecies competition, which result in decline of genetic diversity, and that's real bad news, increased frequency of deletions, retrogenes, premature postdoctorals, etc. And this means that actually these genomes have very low fitness and therefore can be very poorly adapted. In some conditions, we would need to do genetic engineering to rescue these taxa. For people which do in situ conservation, this means that they have to increasingly take attention instead to think about species conservation by simply randomly sampled individuals to put in their conservation. It is important to think about how we conserve the genetic diversity. This means all the individuals we try to take into in situ conservation measurements need to be characterized genetically. A lot of effort and data needed. I could also now talk about genomics in the context of restoration ecology. Actually, I would recommend you to read one review paper which was published a couple of years ago by Breda et al. In Nature Reviews, which actually pointed out that genomics has various aspects how they help restoration ecology. One side is to look to genotypes of the organisms you want to use in your restoration. Genomics can be avoided, could produce a much more sensitive, faster sensitive system to assess, monitor the restoration effects And in some extreme cases, you may actually think about genome editing of your taxa to actually increase the resistance to the change of the environment. Especially if you want to think about restoration on heavy metal depositions, also metal effects, etc. In a very recent publication, Harald et al. Publishing Biological Conservation actually pointed out, despite the genomic tools are there, we see a huge gap between what we say called application gaps in biodiversity, of using genomics in biodiversity ecosystem service management. We need really to take actually much more efforts to really implement this methodology in our efforts. And by that one, I want to take on one paper by Fiedler et al. These are the guys which are behind the California Conservation Genomics project, which is a very nice example for a very wide visionary project thinking genomics and conservation at the same time. The paper may be missed by many conservation biologists because it's published in the Journal of Heretical. Journals are typically read by people working on genes and genomics, population genetics. But they actually use a very nice title, Sizing the Moment, the opportunity and relevance of the California Conservation Genomics project to state and federal conservation policies. And I think they really hit the point home. We need to think about how conservation economics will be implemented in conservation policies in the future. Therefore, I slowly come to the end of my talk and thinking about sizing. Think about the global biodiversity framework, which was only put out a couple of weeks ago. We need to think about reducing the imprint of humans on the earth, especially how we can take away the biodiversity crisis. And here I would argue the genomic tools are one of the key innovations which allows us to take on this challenge. But what do these may need? And I would argue what we need is large-scale conservation genomic projects, which takes species conservation as conservation of genetic diversity. That's exactly the thinking of CPD when they say 90% of the genetic diversity per species. Yes, we have very limited data how much actually species diversity needs per species. Some species have very limited genetic diversity, other species have very large amount of genetic diversity. Therefore, it's actually a challenge to measure and quantify. But basically, such projects are really carefully aligned to the targets of the global biodiversity. We have really to take on the challenge in the genomic phase. If you think about such projects, it could take on a phylogenetic perspective, thinking about sequencing taxa of a phylogenetic lineage, or thinking about a regional or ecosystem focus, where we actually take on a region. Example for a lineage focus would be a project which we initiated a couple of years ago here in Yunnan or here in China, which we call the Terrified Genome Consortium, TGC. Here's an image, shows actually our initial meeting on this with Li Shu as a host of this meeting, which was a couple of years ago, where we want to take on post-genomic lycophytes and fern racers in the post-genomic area. This project would mean that we sequence a lot of the genomes of ferns and lycophytes, which actually happens as we speak. In the context of conservation, maybe it was a more attractive or more productive approach could be regional focus. And I pointed already out 2 projects which actually do that. So Darwin Tree of Life are focused on the British Islands and aim to sequence 70,000 of the species occurring in the British Islands. Whereas the CCGB, as the California Conservation Genomic Project, focus on taxonomy in California. So if you think about globally, we have now 2 regional focus areas which people already have that visionary projects where they try to get most of the genomes of the organisms of concern in these regions sequenced. One is here in the British Islands, Northern Europe, and the other one is in California. Asia lacks so far, as I am aware, a fully proposed project. But you never know which one you overlook. But we should actually think here in Asia very carefully to think about a similar project to do for ourselves. For example, we could think about the Yunnan plant genome conservation project, focus on the rich and unique diversity of this province here in Southwest China, which, for example, when I talk about fern lycophytes, about 10% of the global fern lycophyte diversity is found alone in this province. And actually, that is not unusual. We find these rich numbers and high representation in Yunnan. From a project is also attractive because your climate, your diversity range from the tropical south here in Xishuangbanna towards the alpine habitats in northwestern Yunnan and Shangri-La area. Therefore, huge diversity, huge ecological differentiation, which would be not only from the conservation, but also from the biological research questions would answer. Rather exciting project. Let me finish this talk with 2 pictures. One is this very exciting project in Guizhou province, and one is an image much older from a place which I worked for nearly 10 years, the Natural History Museum in London. Both of these research facilities, both are large-scale research facilities, which actually change how this research in this area have progressed. Both have also in common that a single individual had a vision driving in the context of Natural History Museum. This was a collector who was collecting as much information about biodiversity, in this case very broadly biodiversity of organisms, plants, animals, but also humans. And when he got engaged, he said, I give this collection to the government, but the government needs to build a museum. This is the foundation of 2 museums now, but also a museum would be called the British Museum, which then later on, because based on the size, was built in the British Museum, in the Natural History Museum. And this museum, Natural History Museum, similar to Royal Botanical Gardens, Kew and similar institutions, shaped for a long time how large-scale research in biodiversity has been carried out. Now we are in a new century. We are starting now in the genomics, but we need to think about similar endeavor, how we get large-scale efforts for genome information about the tree of life and the regional biodiversity carried out, and how this information gets provided to people doing conservation of biodiversity. We have to move forward and think very carefully how we take that one. We have to science this now. We're running out of time on one hand, many species have rapid declines of the population size, and this means of the genetic diversity. At the same time, the publication of the global biodiversity framework gives us the right framework and targets to convince people that this is now time to get carried out. At the end of this, I want to point out one thing why I say we're running out of time. Many species experience decreased population size. This means a decrease of genetic diversity. Therefore, this goal to maintain 90% of the genetic diversity in these species will be increasingly difficult. But at the same time, decrease of genetic diversity means enhanced vulnerability to environmental change. The first thing will be more difficult to maintain the species. Let's recover. And with that one, I want to I talked about Charles Darwin very much at the beginning that he has very, very birthday. And I want to take one of his statements, which is very important in our context, back to you. It is not the strongest of the species, nor the most intelligent that survive. It is the one that is most adaptable to change. Humans are very adaptable to change. And we as biodiversity conservation biologists, we need now to use this adaptability to take our new methodology, our new insights by using genomics to conserve biodiversity, to reduce our footprint. Thank you very much for your attention. And I hope I was able to convince you that genomics is one of the key technologies to achieve the United Nations goal, harmony with nature. Thank you very much.